Hi, I'm Dial Holton, Microcalorimetry Product Manager at TA Instruments. I'd like to welcome you and thank you for joining us in today's webinar. During the webinar, you can customize your viewing interface by arranging the windows to your liking. You can resize the video and slide windows independently from one another by clicking and dragging the bottom right-hand corner of each window. If you get disconnected at any time, please use the instructions you received to log back in. You can access several different content windows by clicking the widgets at the bottom of the screen. These include speaker bios, additional file downloads, and the Q&A tool. If you need help, click the question mark widget. Please ask any questions you may have at any time during this presentation by submitting them through the Q&A window. We will answer as many of these as possible at the end of the webinar. Today, I would like to welcome Olivier Bressant to our webinar series. Dr. Bressant earned his master's degree in microbiology, mycology, and the biology of population zoology from the University of Lausanne. He then went on to earn his PhD from the University of Neuchâtel with his dissertation entitled Bacterial Carbatogenesis related to the oxalate carbonate biogeochemical cycle. Olivier's postdoctoral work has included a postdoc position at the University of Connecticut with Professor Peter Vischer, followed by an assistant professor position within the Center of Integrated Geosciences at the University of Connecticut. In 2012, he joined the Laboratory of Biomechanics and Biochemistry at the University of Basel in Switzerland as an associate researcher, where his research has been focused on biological microcalorimetry of biofilms. For the last 5 years, his research has included work on biofilms and biomaterial research, detection of infections, mostly in urology, and the development of rapid drug susceptibility testing using isothermal microcalorimetry. In the last 4 years alone, his work in his lab and with his collaborators has resulted in over 40 peer-reviewed publications, and for the past year, he has also served as the editor for the Journal of Applied Microbiology and the Asian Pacific Journal of Microbiology Research. The title of his presentation today is The Use of Isothermal Microcalorimetry in Urology. Olivier.
Thank you. Hello. Today I'm going to talk about the use of isothermal microcalorimetry in urology and more generally in microbiology and clinical microbiology. And this talk is going to be mostly an overview of the possibilities of microcalorimetry in these three fields. And it will really give an overview and not go that deep in details. So the first thing I would like is that you forget what you know about microcalorimeters and calorimeters in general. We all have been working with different calorimeters during the physics or the chemistry labs, but the calorimeters that we're talking today are absolutely not the same. That's really sensitive and extremely helpful instruments. So what are the advantages of isothermal microcalorimetry for microbiologists and biologists in general? Well, the first advantage is mainly the sensitivity of these techniques. Calorimetry is an extremely sensitive tool that can measure heat production in the range of 20 to 200 nanowatts. And of course, 20 or 200 nanowatts does not tell you that much about what kind of biological process can be recorded. But just to give you a hint, we all know that cells which are provided nutrients and good growth condition will produce more cells and biomass. And basically, this biomass can be measured during protein assay, during DNA assay, or measuring sugars. Cells growing are not only producing biomass, but they're also producing metabolic byproduct. This byproduct can be nitrate, can be sulfides, can be lactic acid, can be also carbon dioxide. And all that you can also measure. But when you consider the overall reactions of bacteria consuming nutrients and producing more biomass and more byproducts, the overall reaction releases a fair amount of heat. And that's exactly what microcalorimetry measures. Now, to give you an idea of how many cells are needed to make an experiment using isothermal microcalorimetry, here is a table that basically lists the specific heat production rate per cell and basically the amount of cells which are needed to get a significant signal in an isothermal microcalorimeter. In this case, I would like you to focus on two specific values, which are the extreme. And this shows that to make a calorimetry measurements, you need from a few hundreds to a few million cells, depending how much heat they can release during their metabolic activity. Now, I would like to go through comparison with other commonly used techniques by microbiologists. Most microbiologists use optical density and use turbidimetry to measure cell growth and number of cells in a culture. So in this case, we made a very simple calculations, and we assume that detectable value in, say, for a spectrophotometer for optical density 600, 0.01 unit, is pretty much 1.5 10 to the 7 bacteria. In this case, it's quite clear that it's at least 2 orders of magnitudes lower sensitivity than a microcalorimeter. So first, calorimetry is much more sensitive. And if you now look at yeast, for example, yeast are detected a bit easier by turbidimetry, but still, it's still 100 of magnitudes lower compared to the tables that you have seen before. Now, if you go to specific assay, and one common assay that many people use is a protein assay, in this case, the bisynchronic acid assay. And it's useful to measure up to 0.5 micrograms of protein per milliliter. What this represents is a really low amount of cells. And again, if you look at the results, it's better than turbidimetry, but still not in the range of what calorimetry can do. Now, another really important aspect for me as a biologist is the simplicity of the measurement. Isothermal microcalorimetry is a label-free, passive, and real-time technique. And one thing is it reduces the use. Since it's label-free, you don't need to use specific, let's say, GFP-labeled organisms or any other GMOs. You don't need to use specific substrate to monitor a specific reaction. For example, you don't need to have fluorescent labels or even radioactive labels on your substrates. And since it's passive measurement, you don't need to withdraw a part of your culture every time you make a measurement. So there is a much less significant biosafety hazard by repeated handling of the culture, since you only put your sample in the calorimeter once and let your measurement run. Now, in the same idea of this simplicity, calorimetry can accommodate every type of samples. You can use solid, you can use liquid, even gaseous samples can be put inside the vials. In this case, since calorimetry only measures heat, any type of samples that your imagination can think about can fit in a calorimeter vial, except for the size. That's the only thing that is, let's say, limiting in isothermal microcalorimetry. So you can have liquid medium. We also did quite a lot of experiments using solid medium, but you can imagine having different materials, for example, implant materials, or even soils. But one important question remains. How do we translate calorimetry data, especially isothermal microcalorimetry data, into data that are meaningful for microbiologists? And that's the next section of this talk will be dedicated to show you how calorimetry data can be interpreted and how we can make sense of this data in terms of microbiology. So the first thing is I would like to tell you is the significance of the heat flow. The heat flow is basically the amount of heat which is produced per unit of time. It's also sometimes called the thermal power. And in biology, the heat flow mainly results from the metabolic activity. And the heat flow can be considered as a proxy for this metabolic activity. If you want to compare it to assays that are commonly used by microbiologists, well, you can compare it to the TTC assay or any XTT, MTT, or Alamar Blue assay, which are metabolic assay. So what you can see in the figure here is just a calorimetric measurements of the growth of Mycobacterium smegmatis, it's the black plain red line, and the parallel measurement done with the TTC assay to measure the metabolic activity of the same culture over time and get the same results. And as you see, both curves are quite well correlated. The peak is at the same place, and the rise of the curve is pretty much the same. Now, it does not look really like a growth curve, but if you integrate the heat flow over time, then you get the accumulated heat over time curve. And this integrations is pretty much the same as in microbiology. The more product you consume, the more cells that you can produce. So in this case, the heat over time curve can be considered as a proxy for the growth curve. Whether you consider optical density measurements or cell count or even byproduct measurement. So the next slide, this slide, will show you an example of that. So here you can see a growing culture of Pseudomonas aeruginosa in liquid medium containing nitrates. And as you know, Pseudomonas aeruginosa can use nitrate instead of oxygen as terminal electron acceptor. So in this case, you have the heat over time curve, the black line, and you have the optical density measurements in red that fits quite well with the curve. And of course, you have the nitrite that is released from the nitrate reduction, which also fits this curve. So the take-home message here is really that if you monitor things with microcalorimetry, what happens is you get the same things as you would get with traditional assays, either optical density, protein measurements, or in this case, nitrite measurements. So since microcalorimetry data can be used as proxy for microbiology data, it's quite clear that you can quantify process exactly in the same way that you would do it for microbiology curves. In this case, for example, it's easy to use the heat and to apply the same growth models that are applied for microbiology, so simple exponential growth models in this case, except that you replace a number of bacteria by the heat that has been produced over time. And when you apply this, you find exactly the same growth rate. And that I will go a bit more in details in some examples later. You can also use the heat flow to calculate the growth rate. It's sometimes a bit more complicated because the heat flow curve can be a bit more bumpy. But since the integrals of an exponential is also an exponential, the calculation of growth rate should be the same. It's just for technical reason, it's always much easier to use the heat than to use the heat flow. You don't need to remain and to still using a really simple exponential model. Now, in a lot of fields in microbiology, there are plenty of models that exist. For example, you can use logistic model, you can use Gompertz models, and all these models can provide you even more information than these simple models. And I will now go in details a bit more, looking at the Richards growth model. In the Richards growth model, you will find exactly the same parameters that you would find in microbiology. In fact, it is a microbiological model. You will find A, which is the maximum growth that you can expect from a culture. If you translate that into microcalorimetry wording, this becomes the total heat that has been produced. You find the maximum growth rate, mu max, that is exactly the same as in microbiology. And finally, you find lambda. Lambda is the lag phase duration, so how long it takes until you get exponential growth phase. And that is all nice microbiological parameters that you can measure and that are helpful. Lambda is usually a proxy for the bacterial load in one sample. The growth rate tells you how fast your culture grows, and the maximum heat produced tells you how much growth the media can support. In this model, you have an additional parameter, nu, which is a small v sign, which is a curve shape factor. A lot of models include such curve shape factor because in calorimetry data, you don't always have symmetrical peaks. You might have asymmetrical peaks, And such curve shape factors are extremely helpful to get much better fitting and much better growth rate and lambda approximations. But calorimetry is also really a chemistry and a physics tool, and we can use it to monitor really specific process like substrate consumption rate. And when you know the reaction which is occurring in your medium, for example, in this case, I would like to focus on calcium oxalate degradation by bacteria, and you have the equation of oxalate oxidation with oxygen. This reaction has a very poor biomass yield, so biomass can be neglected in this equation. And we know exactly the enthalpy of formation of the product, so it's easy to calculate the enthalpy of reactions. And since 1 microwatt is equal to 1 microjoule per second, then it is easy to calculate from the calorimetry data how much consumption of oxalate occurs through a specific amount of time. So in this case, when we put everything together, we can look at the difference between different oxalate degrading bacteria. It's usually a very difficult tool because all these bacteria belong to different genders. And the fact is, it's quite difficult to compare them. Also, looking at calcium oxalate degradation in medium is quite difficult because investigating a solid dissolution in a solid medium is really a pain in microbiology. But it's still possible. With calorimetry, it's easy. You prepare your medium, you inoculate your bacteria, and you get a nice growth curve that you can fit with a model, in this case a Richards model. But from the peak of the curve that you see in panel A, basically you can get the value here and translate that directly in an oxalate consumption rate in micromoles per hours. And in addition to that, basically you get the growth rate and you get all the other factors. So here there is another take-home message which is really important. Out of one single microcalorimetry measurement, you can get a lot of data. You can get the microbiology data, you can get the chemistry data, and you can get plenty of informations which you would not get from one classical culture. So now, going even more in details and closer to the clinics, what use of isothermal microcalorimetry can be made in clinical microbiology and in urology? And there are three main aspects to that. Of course, we can take advantage of the sensitivity of the microcalorimeters to detect infections. Since only a few hundred or a few thousand cells are necessary to get a signal, it's extremely easy to put a sample in medium and just to wait until some heat is produced. And then you get a really quick detection of infections. Another aspect is drug susceptibility testing. In many cases now, it becomes really important in the clinic to get rapid drug susceptibility testing to know exactly what antibiotics can be administered to a patient or can be efficient in this or this specific context. And finally, isothermal microcalorimetry is a great tool in research. The reason is simple. Again, I come back to the take-home message that we had before. With one measurement, you can get more than one information. You would get growth rate, you can get bacterial load, and you can get metabolic activity. So now I'm going to go through these 3 points step by step and in more details. First, I would like to talk about detection of infections. In this case, rapid detection of urinary tract infections. Urinary tract infections are quite important in the clinics. It's the second most common infection just after respiratory infections and cold. So that's quite an important thing in all microbiology lab in hospitals. The issue is most of the samples, in fact, they come out being negative. So no growth in the samples. And last thing, there is a very few numbers of pathogens that represent most of the infection. So it's really a simple model to use microcalorimetry and to get a lot of data quite fast. So we wanted to test how fast can be isothermal calorimetry in detecting these urinary tract infections. And we made a really simple experiment where we used cultures of the 4 pathogens that you have seen before, the bacteria, and we added inoculums of this bacterial culture into filter sterilized urine. We did put everything in a microcalorimetric vial that we sealed and introduced in the calorimeter. And then what we did is we waited until we got a positive signal and we let the calorimeter run for 24 hours just to have a complete curve. So the results were quite clear. As you can see on all these figures for E. coli, for Enterococcus faecalis, for Proteus mirabilis, and for Staph aureus, detection time is quite short. Between 4 to 6, even 9 hours sometimes are necessary to get detection. This is quite short if you compare it to a traditional culture where you would plate your samples and wait 24 hours and see if any growth had occurred. So here in 4 hours we can deliver results and make sure that there is an infection or not. Then you can see on this graph that if you decrease the inoculum from 10 to 5 bacteria to 10 to 3 or even 10 bacteria, then you shift the curve to the right. And this shift is exactly what I told you before with the duration of the lag phase that can be calculated with different models. So the less inoculum you have, of course, the longer it takes until you get exponential growth and a high signal in the calorimeter. Now, there is still one issue remaining. Urine is not the same between patients, and even in one single patient varies over time. So the composition can be quite different in the morning and the evening and between 2 different persons. So what we wanted to do is we do exactly the same experiment in a standard medium, artificial urine that is purely mineral medium in this case. And what we did, in fact, is we did exactly the same measurement in artificial urine and get exactly the same parameter out. And in this case, we got pretty much the same results. I will come back in the next slide and show you what the results were. But you get pretty much the same curves, pretty much the same results, and again, detection between 4 and 9 hours, which is extremely good compared to conventional methods, which takes at least 16 to 24 hours. So now, just take a look at the results now. You have these big tables, and we artificially set the detection limit for a positive infection at 10 microwatts. That's a very, very conservative value because the detection limit of the calorimeter, as we have said in the first slides, is around 200 nanowatts. So it's 50 times higher than the detection limit. But it's very conservative, and it's important in the medical field to remain conservative. Now I would like you to focus on this value here. 10 to the 5 bacteria per milliliter is considered as a threshold between urinary tract infections and no urinary tract infections. So in this case, you see that detection is extremely fast. In filter sterilized urine, you only need, let's say, 4 to 6 hours to get detections. In artificial urine, you need around 3.5 and 9 hours to get detection. Again, that's much, much better than what we can do with traditional culture. But is this the only thing that we should take out of such a measurement? And of course, the answer is no. Because with calorimetry, you get real-time measurement and you get this curve which contains a lot of information. It's not just detection or not detections. You get plenty of additional information. So in this case, you can get insights into the pathogenicity of a specific strains that provoke this urinary tract infection. And in this case, we calculated the growth rate for all the different strains that we tested. And of course, from that, we calculated the doubling time in hours. And it's quite important because most of urologists would consider that the fast-growing strains are able to maintain themselves in the urinary tract, as the slow-growing bugs would be flushed when you go peeing, for example. But the important thing here is that calorimetry data are exactly similar to what you can expect from the literature data. So it's not a vision of the mind to use calorimetry as a tool to get insight in microbiological parameters. If you compare the data that I have outlined here in red, you see that the doubling times that we measured are perfectly in line with the literature value that can be found. And these literature values are extremely difficult to find because growth of bacteria in urine is poorly investigated. And in fact, what you see here is that you can measure things and you can, in fact, get insights into this pathogenicity. And the important other thing is you get quite a good correlation between the measurements which are made in urine or in artificial urine. But again, is this really enough? And the answer would be, of course, no. Rapid detection of infection is a great thing, but it would be even more interesting in the clinic if we were able to rapidly treat these infections. And to treat an infection, then you have to know about about drug susceptibility. And in this case, testing rapid drug susceptibility with calorimetry. So what we wanted to do in the second part of our test with a calorimeter in the field of urology was to compare drug susceptibility testing as it is done in the hospital using the VITEC system, which is an automated system that focuses on either bacterial identification or drug susceptibility testing, and compare with the results of the calorimeter. So can a calorimeter get an accurate drug susceptibility profile, and in how long can it be done? So we focused our effort on one specific case, the case of urosepsis. As you know, septic shock kills a lot of people every year, and urosepsis is basically the beginning of a septic shock. It's defined as a sepsis originating from the urinary tract. There are major advantages in studying urosepsis with calorimetry. One is that urosepsis are pretty much always accompanied by a really high bacterial load in urine. So there is no need for concentration, there is no need, we can use urine as is. The other thing is, in most cases as well, there is a single pathogen that can be isolated from blood, if it's isolated from blood, and from urine. And this pathogen is the same pretty much all the time. So then the idea is to use a urine samples and add a few antibiotics to it, and finally get a drug susceptibility pattern. So I will go directly to the results. And in this case, after 7 hours, we were able to provide the drug susceptibility pattern. So what you see on this graph is a bit complicated, and I will try to guide you through it. First, after getting the calorimetry curve, what we did is we calculated the growth rate, the lag phase, and the total heat produced. And from these values, we calculated an inhibition index. Basically, this inhibition index is the ratio between how much growth was achieved by the control over how much growth was achieved by the samples treated with antibiotics. And we got an inhibition index that ranged from 100% to 0%. And we compared it from the susceptibility determined by the VITEC system. In this case, you have the name of the antibiotics at the bottom of the graph, with S for susceptible and R for resistant. So as you can see here, there are really clear-cut data for most of the antibiotics. So the determination of drug resistance or drug susceptible is extremely good, at least for the 3 antibiotics on the top of this graph, and still quite good for the antibiotics at the bottom of this graph. As you can see, the 3 antibiotics at the bottom of this graph are mostly beta-lactams. And the fact is, the mode of action of beta-lactams makes that they are slightly slower acting than, for example, fluoroquinolones like ciprofloxacin or penem like ertapenem, for example. And since they are slow acting, the bacteria still grow a bit in the medium before it's completely inhibited, which explains that we measure a slightly lower inhibition ratio. But what is important in this graph is really let's say, the cutoff values, that there is still a big difference between resistant and susceptible strains. Now, just to summarize this data, I would say that there is a good agreement between the Vitek results and the calorimetry. We also did classical culture measurements, and basically the main result is, in most of the case, we have an agreement between the three techniques. And the other really important thing is, in all the tests that we have made, we have only one cases where calorimetry provides a wrong result, sees a resistant strain as susceptible. So that's the take-home message. Calorimetry is at least as accurate as a Vitek system, but much faster. In 7 hours, which includes 30 minutes for sample preparations, 6 hours of calorimetry measurements, and 30 minutes for data treatment, you can get an accurate drug susceptibility profile. And it's not reserved only for urosepsis. It is also applicable to any type of drug resistance. So in this case, we focused also on methicillin-resistant Staphylococcus aureus. And as you can see on the left side, basically susceptible Staphylococcus aureus do not grow as soon as there are antibiotics added. On the other hand, on the right side, you see that even if there is antibiotic in the medium, you can see growth of this Staph aureus, and you see that in 500 minutes, so between, let's say, 6 and 9 hours, you can have accurate results. So rapidly detecting resistance in Staphylococcus aureus. And again, that's a very important bacteria for hospitals and hospital hygiene. Now, I reached the last point of what we can do in urology, and I would like to focus on research, and especially research where calorimetry can make a big, big difference. Research where it's usually quite difficult to investigate growth or investigate metabolic activity with conventional techniques like culture, like microscopy, and so on. So I will focus on 3 different things. The first will be fungal infections, mostly focusing on Candida albicans. The second will be parasitic worms, schistosoma in this case. And finally, I would like to talk a bit about urinary tuberculosis. So first, Candiduria and Candida albicans infections. In most people, Candida albicans is a common inhabitant of your body. And in most of the case, Candiduria is asymptomatic, so that's not really worrisome. But in some case, Candida can make an infection, in fact, a urinary tract infections, and lead to really severe consequences, so kidney loss, big infections, even death sometimes. And one thing is the risk factors are usually known for Candida infections. It's usually related to diabetes, to indwelling catheters, to urinary tract device, and antibiotic use or overuse, also in immune-compromised patient like HIV patient or patient undergoing chemotherapy. But the thing is, even if we know these risk factors, there are very, very few data on Candida relation to glucose, for example. And that's not really surprising because Candida in urine tends to form pseudomycelium. And all these cells which are packed together appears as a single colony when you plate them, or are quite difficult to investigate by microscopy. Nobody is going to count how much cells there is in an aggregate. But again, with calorimetry, you don't really need to do that because you only measure heat and heat flow. And you can get really a quite accurate measurement of the growth rate just by measuring the heat flow, integrating and fitting a model like I've described it before. And now, we can take a look at the influence of glucose on the growth of Candida in urine and in the urinary tract. In this case, we had a small glucose range between 20 and almost 2000 milligram per milliliter, and that corresponds to what you could find in healthy patients, up to 200 milligram per milliliter. In diabetic patients, this value can reach much higher levels, almost 80 grams, per liters of urine. So you see that it can be a lot of glucose and a lot of food for candida in urine, in fact. So in this case, we can see that even if you add tiny amounts of glucose, you will increase the growth rate. On the range that we considered here, adding almost, let's say, 2 grams of glucose per milliliter, 2 milligrams of glucose per grams of urine, will lead to 10 times increase in the growth rate. That's extremely important. That means that basically a yeast will grow 10 times faster. But it's not the only thing that we can do with a calorimeter in this case. We focused a lot on the use of antibiotics. And a thing that happens quite often in candida is after antibiotic treatment, it relapses. And we were interested to know exactly what happens in urine when you put antibiotics and yeast. And one thing that was really striking is you can measure the metabolic activity of really small amount of yeast over time, over pretty much 1 month in this case, when they are under the influence of antibiotics. And as you can see on the bottom panels, the number of cells never goes really high, around 1 million or a few millions per milliliter, extremely low levels. But on the other hand, they're not supposed to grow because there is antibiotic in the medium. But you see that survival takes some energy. And you see that really well with the calorimeter, because in this case, you can see that there is a low but quite regular metabolic activity over the period that we considered. One thing which is really striking here is each time you have a peak in the metabolic activity, you also have a peak in the cell number. So again, it's not a vision of the mind. There is really something happening. There is more metabolic activity and then suddenly there are more cells. Not much, but it occurs. And calorimetry provides a very, very sensitive tool to monitor that without having to go through painful measurements, microscopy, plate count, and so on and so on. Now there is another interesting case for calorimetry, parasitic worms. Drug testing in parasitic worms is quite complicated. In fact, it's not complicated, but it's mostly time demanding. The main technique to measure susceptibility of worms to different anti-worms compound is basically to expose these worms to this compound and then to look in the microscope if they're still moving, if they have some viability, or if they are dead. But mostly the main criteria is, are the worms still moving? Of course, as you can guess, microscopy is extremely time-consuming and subjective. Different people will just consider worms dead or worms alive. Or you might just look in your microscope and don't see the worm moving, and it will move just after you stop watching at it. That happens. Making time series of such measurement is extremely time-demanding and demands a lot of manpower. You need somebody that comes every 2 or 3 hours, look in the microscope, count how much worms are still moving and are still active, in fact. And finally, the results of this testing are quite difficult to interpret because you're never sure that a worm is dead. So that's pretty much it. And of course, microscopy cannot be automatized. So it's, again, quite difficult, and it makes a strong case for the use of calorimetry. And we did several tests with the Tropical Institute in Basel. And basically, what happens is originally, we looked at the signal and thought, wow, there's something wrong with this signal. And of course, when you see such a signal provided by your calorimeter, you're a bit worried. There's a lot of noise in the signal, and you're not sure that you're going to be able to take something out of it. But if you look more carefully at what is happening in your calorimeter, and you look carefully at your signal, you will find that this noise is not a mistake. It contains informations. And that's what I would like to outline right now. If you look at a complete profile of such parasitic worms put in the calorimeter, you will see that there is in fact 2 signals in your results. There is, if you perform a smoothing, there is the smooth line is kind of a proxy for the overall metabolic activity of the worm. So how much heat does he release? But the additional noise, in fact, is linked to the worm motor activity. When the worm moves, there is a slight amount of heat that is released, and there is a small perturbation that is seen by the calorimeter. So now you pretty much see where I'm going. Basically, you don't need to stand in front of a microscope for hours to know if a worm is dead or alive. You just need to put it in the calorimeter and just monitor his motor activity and his overall metabolic activity. And there you have a clear-cut signal where you can see when your worm is moving or when your worm is not moving, and the metabolic activity is low. That means that your worm is dead. And that provides a great tool for testing new anti-schistosomal compound in this case. The last part of this talk will be a focus on urinary tuberculosis. It's one of the most common extrapulmonary manifestations of tuberculosis. It's an extremely slow-growing bacteria and grows even slower in the urinary tract. And a recent study by Russian people has shown that 100% of cases of urinary tuberculosis were misdiagnosed at the first visit. And still, it's slowly growing, but it's very destructive because most of the time it's diagnosed so late that there might be renal failure or testicular destruction in this case. And again, it's such a difficult bacteria to investigate because the time scale for performing an experiment are so long that there are very, very few data. So what we did in this case is pretty much the same setup as you have seen for the urinary tract infections, because in fact urinary tuberculosis is a urinary tract infection. It's just much more slow. And in this case, we added serum in the process to make sure that some bacteria would grow. And the results were quite clear. I let you take a look at the time scales. It's a very, very long time scale. And you see that the fast-growing mycobacteria, which are Mycobacterium smegmatis, which is a common model for Mycobacterium tuberculosis, and Mycobacterium flei, which is also a model for Mycobacterium tuberculosis, they grow quite fast. I mean, fast being in the range of 50 hours in this case. But if you take the slow-growing nasty urinary pathogen like Mycobacterium kansasi and Mycobacterium tuberculosis, you see that first there is really, really little activity, and also that for tuberculosis in plain urine that there is not growth. So we added serum and we did the same experiment, and I'm not going to show you all the graph, just going to show you the results. And I would like you to focus on these values here in red, which are the doubling time of Mycobacterium tuberculosis and Mycobacterium kansasi in urine and urine plus serum. And you see that the doubling time can be in the range of 100 hours. 100 hours is pretty much 4 days. And then you understand that performing an experiment, a long-term experiment with this bacteria becomes quite difficult. And especially if you have to use classical technique and withdraw samples from time to time, this causes an important biosafety concern because you're going to have, during 40, 50 days, to withdraw samples from a culture of a biosafety-free organisms and basically process these samples really carefully. With calorimetry, you can put everything in a single vial and wait during 40 days until you get your results. Something was also striking in this experiment is that you have formations of biofilms in some samples, and urinary tuberculosis seems to be quite enhanced by the formation of biofilm. So I'm going just to highlight the two curves where biofilms were formed. And we have currently no explanation for the formation of biofilms in this culture, but the fact is, when a biofilm forms in the vial, you have a much longer activity and a much higher activity over time. So biofilm formation by mycobacteria seems to be really important and underestimated process that makes these organisms extremely pathogens. And that you can only see on such a time scale and with real-time data using a microcalorimeter. So I have reached the end of this talk, and I just want to outline a few conclusions. And first, it seems obvious to me now that isothermal microcalorimetry is a very versatile tool that allows to study a wide range of biological processes, and especially for me, the one which involves cells from different types or different microorganisms. The sensitivity of isothermal microcalorimetry is usually higher than other commonly used techniques. Microcalorimetry data can be interpreted in most of the cases exactly as microbiological data would be, and thus automation of the process is possible. You can automatize data processing, and then you get a lot of information in not that much time. Finally, in life science and detection of infection, rapid drug susceptibility testing, parasitology, and cancer research could be nice application field for microcalorimetry. So isothermal microcalorimetry is also a very versatile tool for performing research on slow-growing organisms. You have seen the last case of urinary tuberculosis, and I would really emphasize that in this case, calorimetry is an enormous help, because with a few samples, you get the information that would take you forever to gather with conventional techniques. There are many other potential use for isothermal microcalorimetry, and I've really show you only the tip of the iceberg in this presentation. So I engage you to get some more readings and look for specific applications that you might be interested in, but there is tons of things which are possible with a calorimeter. And finally, I would like to acknowledge all the team of Basel, especially Dieter Wirth, Gernot Bonkat, Alexander Bartman, and Dan Daniels, which have been really supportive of use of microcalorimetry in the hospital and in material science, in implantology, and all the students, Daniela Rodriguez, Anna Solokina, Lucas Vernley, and Mary Howell, that have been of great help in this research. And finally, I would just like to remember you that, again, it's only the tip of the iceberg. Much more can be done with calorimetry, and I am happy to answer questions if you have any.
Thank you, Olivier, for that informative presentation on isothermal calorimetry.